So this is the second episode of the second season of Medicrew Masterclasses, and today we are going to discuss about the physiology of learning and memory, and in application we want to learn about the different forms of dementia and their presentation. So the first part of today's session is about the physiology of learning and memory, and the second part will be on the pathophysiology of dementia. Let us start with the first section. So we start with a few questions, and this is your first question. Learning is defined as. The next question. So if you identify this picture and immediately say that this is an apple, what form of memory are you using? The third question. A mother wakes up every time the baby cries during the sleep. So this is a reflex phenomenon. So what is this an example of? How do you explain this physiologically? The fourth question. Retrograde neurotransmitter, that would be neurotransmitter. Retrograde neurotransmitter in long-term potentiation is. And the last question before we start discussion. What does hippocampus store in relation to memory? Okay, so with this we start the discussion on the physiology of learning and memory. We know one characteristic of humans is that we can alter our behavior on the basis of experience. For example, if we touch something and that experience is painful, we'll stop touching that, we'll avoid that object. If we eat something and it is very pleasant to eat, then we will be eating that again and again. So there are certain kinds of positive and negative reinforcements that team up our learning experience. So it is a very intrinsic characteristic of animals, particularly the higher animals, which is their ability to change their behavior on the basis of experience. In order to do this, we need two particular components. One, we need to learn new information. For example, we need to learn that if you touch a pot of boiling water, it will hurt. So that is an experience, that is a fact. And then that has to be remembered for long enough so that next time when a boiling pot of water is placed in front of you, you don't touch that. So the acquisition of the new information is learning, and retention and storage of that is known as memory. So when you learn new information, that process is learning. So learning is essentially acquisition, not necessarily storage. So you may be a short-term learner. You can learn and forget also. But when you learn and remember, that is memory. So the learning is then converted to memory. And this definition also tells you that learning and memory are intimately related to each other. Without learning something, there cannot be any memory because you need a database that you need to remember. And once you remember things, the learning becomes consolidated. So although they are two separate entities, one is the acquisition and the other is the storage, but still they are intimately connected with each other. That is why different types of learning and different types of memory are often classified together, and they actually cannot be separated explicitly. So what are the different types of memory? There are different ways of classifying memory based on the content, based on the duration, like short term, intermediate term, and long term. So there are different ways in which memory can be classified. In today's discussion, we are classifying memory based on its function or content, not necessarily on the duration. Although we will, of course, match it up with the classification based on duration. Memory, broadly, based on what it is about, it can be divided into two forms: declarative or explicit memory and non-declarative or implicit memory. So what is declarative memory and what is non-declarative memory? Declarative memory are essentially facts or events that you remember. Non-declarative memories are essentially the skills that you develop, the intuitions that you develop. It's not something that you can really speak about, but it's something that you can perform. So when you learn to ride a bicycle, then that is an implicit memory. You don't remember to ride a bicycle by 5 steps. You know how to do that. The reflex pathways of the body has learned that technique. But then if I ask you, what was the cost of your bicycle? Or who gifted you the bicycle? Or what are the parts of a bicycle? That is a factual memory or an episodic memory, which is declarative in nature. So whatever we do in our day-to-day life is a combination of declarative and non-declarative phenomena. We are learning subconsciously, we are also consciously remembering information. And that is how certain informations become declarative memory, certain skills become non-declarative memory. This will be more clear as we give examples of each type of memory. Memory, as I told you, can be classified in multiple different ways. So one classification of declarative memory is to classify it into two groups: semantic memory and episodic memory. Semantic memory are crude facts, so they are just informations, whereas episodic memory are experiences that you can remember or revisualize in your life. For example, if you remember the name of a book, the name of its author, or the price of the book, or the name of a person who gifted you the book, these all informations are facts. But when I ask you something which is related to an activity, like how many people were invited to your birthday, who was there beside you when you were cutting the cake, these are episodic memories. So episodic memories are like video photographs that are imprinted in your mind, and you can, if you remember, see it again and again. The semantic memories are crude essential facts. So they essentially answer the question what or who, whereas episodic memory is all about a detailed description of the activity. In both the cases, for storing both semantic and episodic memory, the temporal lobe plays a very important role. The lateral and anterior part of the temporal lobe stores facts, whereas the medial part of the temporal lobe stores the events, that is, episodes. Also important is the hippocampus, which is important for episodic memory, and it also converts short-term memory into long-term memory. So let's take this example. You look at something and you start uttering the names. So you look at an apple and say, this is an edible thing. This is a fruit. This is hanging from a stem. It has seeds inside it. It grows on a tree. It is red in color. It is almost round in shape. It is sweet to taste. These are all semantic memories. Crude factual informations that can be bulleted about an object or a person. Episodic memory is more about when you went to an apple orchard and then you remember who spoke to you, how many people you met. So this whole conversational thing that happened there is the episodic memory. And as I told you, in both these cases, the fundamental questions are, in semantic memory, the activity is I know. So what do you know? When you start answering that, that is your semantic memory. But when I ask you to relive that experience, to describe that in the form of a film or a script, then you start narrating your episodic memory. And both these memories are stored in the temporal lobe and other parts of the brain. So you can easily guess from here that the lateral and anterior temporal cortex is the storage center for semantic memory. It also takes the help of the anterior part of the brain, which is the prefrontal cortex. Whereas the medial temporal lobe, the upper surface of the brain, that is the neocortex, and hippocampus deep into the temporal lobe, these parts store the episodic memory or the events. After the declarative memory, which is either facts or events, we now come into non-declarative or implicit memory. The implicit memories are mostly skills that you develop. They are not exactly facts that you can talk about, but they are things that you can perform. And we will discuss separately about all these different types. One very common implicit memory is a procedure, like riding a bicycle or playing a violin. These are skills that we learn. And these skills are stored in those parts of the body that regulate our movement. So quite understandably, that will be in the motor cortex, the coordinating part of the body, that is cerebellum, and the planning part of the body, that is basal ganglia, that is striatum. So the planning, coordination and activity, all these things are all put together in proper position through striatum, cerebellum and motor cortex. Striatum helps in planning, cerebellum in learning the coordination, and motor cortex executes the plan. And these three areas, therefore, remember how to do something. That is the procedural skill. Another very important skill is known as priming, which is developing intuitions. And I will come into details of priming later. Classical conditioning, like the Pavlov's experience, where a dog was attuned to a ringing bell and food, this kind of associative learning occur in the amygdala and cerebellum. Amygdala, by the way, is a center for emotional response. And non-associative learning are two special types of learning that involve reflex pathways. So let us go one by one into these different types of learning and memory. So first, talking about the procedural memory. The procedural memory is all about how to do something. So when you say, I know how to do this, you are essentially talking about your procedural memory. So how to write, how to read, how to ride a bicycle, how to swim, how to cook, how to behave, all these are learned traits, and these are all procedural memories that are impregnated into the motor circuit of the brain. The cerebellum, the basal ganglia, that is striatum, and the motor cortex. So in this way, these three areas coordinate to remember the procedural memory. Priming is a very interesting fact. So it's a question to all of you. Read this and fill up the gap immediately before I count 5. So the time starts now. 1, 2, 3, 4, 5. So see, most of the answers here are grape. Green is something that you have to write consciously, right? On the other hand, had the question been more like this, and this test is not applied in this way. It is a wrong way to apply this test. This test should be ideally applied into blinded groups, where one group is getting only one card and not the other card. And then statistical evaluation should be done to see the majority of the responses. So you see, when your brain is thinking of fruits, GR elicits a response like grapes. When your brain is made to think about colors, GR elicits a response like green. While it's not always necessary, but this is usually the fact, and that actually is helpful from an evolutionary point of view because it helps us to be in a context. We remain in the context of the situation by doing this. This kind of an intuitive understanding of something is known as priming. Another example of priming you can easily guess. When most people will be given this towel, shower, shampoo, and dash, what do you think the answer would be mostly? The answer will be mostly soap. Because we are talking of toiletries. Whereas if another group is given this kind of a jargon, then maybe most of them will be writing soup because we are talking in the context of food. Again, this is not mandatory that the answer has to be this, but in majority of the cases, the answers would be like this. And this is known as priming. It's your intuitional ability to extrapolate a situation and find the next. This priming is a very advanced function of the human brain. And this is mediated by the outermost layer of the human brain known as the neocortex. Then comes associative learning, and associative learning is a conditioned learning. So what is a conditioned learning? A conditioned learning or associative learning means two stimuli are paired with each other. And when one stimulus stimulates your body, the other stimulus gets linked to that. In the classical conditioning that we have all learnt, it was all about a bell ringing and a dog was given food. So initially the two stimuli were different. But as the two stimuli started firing the neurons together, later on a mere whistle or a bell will be causing salivation in the dog. Operant conditioning, on the other hand, is a heat and trial method. In this case, you do not pair the stimulus. Suppose in the whole cage, there is one button or there is one lever as shown here, which the rat can use. And when the rat presses this, food comes out of a hole. Now the rat was never trained to do that. Nor did you train the rat or in a way instruct the rat to do this. The rat was playing around. Accidentally, when one day the lever was pressed, food came out. Then more and more accidents started happening, which led to an observation. And the rat started understanding that each time I press this button, a pleasurable stimulus comes out. And so the rat will repeat that again and again. This is known as operant condition. In real life also, similar things happen. For example, if you teach a child to go to the toilet and keep the hose pipe or keep the tap open, then the sound of the water and the sensation of micturition gets conditioned together. Next time, if the child has a problem in micturition, if you just open the tap with the sound of the water, the urge of micturition will develop. This is classical conditioning. On the other hand, suppose if a child micturates in a classroom, the child gets scolded. The scolding acts as a negative reinforcement. And if a child raises the hand and asks for permission to go to the toilet, the child is given, suppose, a good compliment, which is a positive reward or a good stimulus. So the child by heat and trial learns what is right and what is wrong through reward and punishment phenomena. Understands that micturition inside the class is not acceptable, whereas raising the hand and asking for permission is acceptable. This kind of social learning behavior, similar to the rat pressing the lever and learning that this is a positive stimulus, is an operant conditioning. What is common between the two is, in both cases, one stimulus and one response are getting tagged together. And this kind of learning is known as associative learning. Associative learning is a reflex phenomenon, and this happens through amygdala and cerebellum. The amygdala mediates the emotional component of reward and punishment, and cerebellum moderates probably the motor component of this associative learning. Finally, we come to non-associative learning. Now, there are different kinds of non-associative learning. Two common types of non-associative learning are sensitization and habituation. So, what is sensitization and what is habituation? Let us understand. When a stimulus is repeated again and again, we either become highly sensitive to that stimulus and react even more vigorously, or we become numb to that stimulus and stop responding. When we become overtly sensitive to the stimulus, that is called sensitization. And when we start ignoring that stimulus, that is known as habituation. So let me give you an example of this. Suppose there is a mother, and the mother is lying down on a bed by the side of a busy street. So there are cars honking, there is a lot of noise, and the mother learns to sleep in that. So the sensory system ignores the sound of the car. But that doesn't mean that the whole auditory system shuts down. Within all this noise, when the child is crying, the mother wakes up. Because to the mother, a child crying is a noxious stimulus. It is not a pleasant stimulus. It indicates danger. And when a stimulus is associated with danger, sensitization happens. So at a biological level, how does sensitization happen? Suppose there is a sensory stimulus coming, like hearing the cry, and the response will be waking up. When the mind or the brain perceives the stimulus as dangerous, then it causes firing of another neuron known as the facilitator neuron. And the facilitator neuron releases serotonin. This serotonin causes enhanced release of neurotransmitter and exaggerates the response. So this is like an exaggerated startle response. A common example is phobia to something very specific. Like suppose you have a phobia to cockroaches or phobia to snake. So in that case, because it's a noxious or unpleasant stimulus, it is not that if you look at the snake again and again, immediately you will stop fearing the snake. Instead, each time you see the snake or hear the hissing of a snake, you get more alert. This is because of sensitization. On the other hand, habituation is just the opposite of this. You start to ignore many things. Like you ignore the noise of the surrounding when you are attentively watching a movie. You ignore the noise outside your room when you are sleeping. You ignore the smell of a particular perfume by sensory adaptation when the perfume has stayed for a longer time. So these are all examples of habituation. Although the mechanism of sensitization is clear, that it is caused by a serotonergic neuron, the mechanism of habituation is not very clear. We know that the response gradually decreases, but how that decreases is not very clear. One important model was proposed by a scientist called Sokolov, and that's a very interesting model. What Sokolov proposed is that when you experience a particular type of stimulus, brain makes a copy of that stimulus. And if the stimulus is not perceived to be very dangerous, then each time the stimulus comes, the brain compares. Is the new stimulus same as before or different from before? If it is same as before, the brain shuts down the response. And this kind of mechanism prevents further response to a common stimulus. In the sensitization, we saw that the serotonin acts on the neuron and causes further increase in calcium and exocytosis. The opposite happens in habituation. Although the precise mechanism is not clear, but there is a decrease in calcium in the neuron, so release of neurotransmitter gradually decreases. So these are the different forms of learning. And finally, we come to a mechanism called long-term potentiation. It's a very common idea that if you read something again and again, you remember that better. So how is it that reading something again and again or doing an activity or practicing something again and again makes you perfect on that? This kind of a long-term learning can be explained by a phenomenon known as long-term potentiation. The mechanism is essentially simple. When a particular neuron is stimulating the next neuron again and again because of repeated stimulus, a lot of calcium goes inside the cell. This large number of calcium causes some fundamental changes in the function and the structure of the neuron. It increases the number of receptors, it increases the sensitivity of the receptors, and it probably also helps in formation of new branches of the neuron or new synapses. It also releases nitric oxide, which goes back to the presynaptic neuron as a retrograde or oppositely moving neurotransmitter. And this nitric oxide causes increase in more neurotransmitter in the previous neuron. So ultimately, there is strengthening of the synapse, which is known as long-term potentiation. And this literally translates into the fact that practice will make you more and more perfect. That is why repeating one activity mindfully again and again will ultimately cause consolidation of the memory. And this is through long-term potentiation. During long-term potentiation, new synapses form. So the synaptic strength and activity increases. Also, the number of connection of the synapses increases. So this is an actual photomicrograph of two neurons forming contact. And you can see when they were stimulated repeatedly, how the number of axons and dendrons are multiplying in leaps and folds and new connections are forming. The more the connection, lesser will be the resistance of electricity flow, and therefore there will be a better transmission of the impulse and faster execution of the activity. So this is how synaptic plasticity occur through the long-term potentiation. So let us quickly revise what we just now discussed, because it is a bit complicated thing. We learned that a characteristic of animals and humans is to alter behavior on the basis of experience. Learning is the acquisition of this information, and memory is the storage of this information. Memory can be declarative or non-declarative. Declarative or explicit memory can be facts or semantic memory, events or episodic memory. Semantic memory and episodic memory are stored in the temporal lobe. Additionally, semantic memory is stored in the prefrontal cortex and episodic memory in the hippocampus and neocortex. Non-declarative or implicit memory can include procedural memory, which means how to do something, and this is stored mostly in the motor parts of the brain, the striatum, cerebellum, and motor cortex. Priming is an intuitive learning where you guess what comes next. And this priming is mediated by a highly advanced part of the brain known as neocortex. Associative learning is where a stimulus and response are paired together and they start getting linked, which can be classical and operant conditioning and is mediated by the emotional center, amygdala, and the motor center, cerebellum. Non-associative learnings include sensitization and habituation, and they operate through reflex pathways. Sensitization is because of a facilitator neuron which releases serotonin and increases calcium, whereas habituation is due to some unexplained mechanism that decreases calcium. Long-term potentiation consolidates memory, and it explains why repeated practice makes us perfect. So up to this, is this clear to everyone? It is the long-term potentiation that leads to neuroplasticity. So long-term potentiation is the cause and neuroplasticity is the effect. So let us start answering the questions that we started this. Learning is defined as wonderful. Learning is defined as acquisition of information. So what kind of memory are you using when you call this an apple? So since this is just a fact, name of something, so this is a semantic memory. When a mother wakes up from sleep with her baby crying, what is that an example of? Wonderful. This is an example of sensitization. Retrograde neurotransmitter. What is an example of that? Very good. The answer is nitric oxide. And finally, what does hippocampus store? Yes, hippocampus stores episodic memory. So with this we end the first section, that is the basic physiology of learning and memory. You can easily understand that there will be many questions and I will take the questions at the end of the session. So please keep your questions with you. Once the session ends, I'll start taking questions. The next part we start with is dementia. So let us start with a few questions. A patient with Alzheimer's disease will frequently complain that he is forgetting everything. Is it true or false? Next question. A potentially reversible cause of dementia is? Next. Example of multifocal dementia is? That is a dementia that affects multiple parts of the body or the brain. Next. Now look at this histological picture. This is a histological picture of the brain stained with silver stain. And you can see there are some small black areas and some large black areas. I have labeled the large black area. What is this large black area out of these four? And the last question before we start the final discussion. Treatment of Alzheimer's disease includes? Okay, so with this, let us start the second part of our discussion, that is dementia. Dementia is defined as an acquired impairment of memory. That is, the memory loss should be acquired. It is not from birth that the memory did not form. Instead, the memory formed and then it was lost. So it is an acquired impairment of memory. But that is not sufficient to call it dementia, because loss of memory is amnesia. Along with the loss of memory, there should be a global cognitive and intellectual decline. The overall intellectual function has to decrease. Otherwise, that cannot be called dementia. So in dementia, one criterion is the acquired impairment of memory. But any one out of these four should also be present. Aphasia, which we have discussed in the previous episode, which is a cortical language dysfunction. Apraxia, that is inability to perform some activity. Agnosia, that is inability to remember some object or thing. And disturbance in executive function, that is when you instruct the person to do something, the person cannot make sense of that. When any one of these at least is present along with an impairment of memory, then the condition is called dementia. In dementia, what is very interesting is that the patient does not even remember that the patient is forgetting. So the patient will never tell you that I am forgetting things. If a person is absent-minded due to depression or due to any other kind of stress, then the person might most likely tell you that I am forgetting things. But a person really suffering from dementia will never come up to you saying that they are forgetting things. Instead, they will insist that they have not forgotten anything. It is you who is falsely charging them of forgetting. They will instead become very angry. They will think that you are an enemy and therefore become paranoid. So that is a very important characteristic of dementia. That is also the reason you should never confront a person with dementia and make that person understand that the person is forgetting. Always be cooperative and explain that that person is right, because that person will never understand that that person is not right. So this is how we differentiate dementia from other forgetfulness related conditions. So true dementia is never characterized by the patient saying that I have forgotten things. There are various causes of dementia. Some of them are reversible, meaning that there is some treatment for that if done in the right time. And there are some causes that are irreversible, that damages the brain permanently. And there are no good treatment for that. So the potentially reversible causes, as you can see, are hypothyroidism. Severe hypothyroidism can cause dementia. But if it is identified early, you can start treatment with thyroid hormones and the person will get back to normal life. Subdural hematoma, that is a blood clot in the brain, can press the brain and therefore impair the function. If a proper neurosurgery is done in right time, then subdural hematoma or tumors can easily be cured or relieved. Inflammation of the brain, known as cerebrovasculitis, can also cause dementia. But if anti-inflammatory drugs and glucocorticoids are used in the right time, in the right dose, then of course it can be largely prevented. In normal pressure hydrocephalus, the CSF cavity enlarges. So if you can do a surgery to reduce the size of the cavity, again that can be partly reversed. If dementia is caused by drugs that cause forgetfulness, like the antihistaminics, or drugs that cause excessive drowsiness, like the benzodiazepines, you can modify the drugs or withdraw the drugs whenever possible and therefore reverse the dementia. HIV is a treatable condition. So when identified very early, AIDS-related dementia can be prevented. Vitamin B12 deficiency affects the neurons. So it causes peripheral neuropathy, it causes damage to the spinal cord, and in the late stage, it causes dementia. But if it can be identified early and supplementation of B12 can be done, then it becomes a potentially reversible cause. That is why it is very important that in any case of dementia, you find out the reversible causes. Because if you can identify a reversible cause, the patient can be saved. However, there are some causes which cannot be totally reversed. These include degenerative diseases like Alzheimer's, diseases where strokes damage the brain like vascular dementia, diseases where abnormal proteins deposit in the brain like dementia with Lewy body or dementia in Parkinson's disease, diseases where there is atrophy of the frontal lobe, so decision making ability goes away, and the person behaves in a weird way, like the frontotemporal dementia. Prion diseases where the brain undergoes spongiform decomposition or degeneration, known as the Creutzfeldt-Jakob disease, which has no good treatment till date. And late stages of neurosyphilis when no treatment can be done. Of course, in the early stages, you can treat with penicillin. These causes, the reversible causes as well as the irreversible causes, some of them affect the cortex, some affect the subcortical structures, some affect both, and some affect multiple parts of the brain. Accordingly, anatomically, we can classify dementia into some types. Cortical dementias include frontotemporal dementia. Posteroparietal dementia means the posterior and parietal part of the brain are affected. Example is Alzheimer's disease. So these are all examples of cortical dementia. The lower part of the brain, like the basal ganglia, etc., are largely damaged in Parkinson's disease, Huntington's chorea, Wilson's disease, HIV dementia, and normal pressure hydrocephalus. Both cortex and subcortical areas are damaged in dementia with Lewy bodies, and multiple parts of the brain diffusely are damaged in Creutzfeldt-Jakob disease. So a cortical subcortical dementia is the Lewy body dementia, whereas multifocal dementia is the Creutzfeldt-Jakob disease. It is impossible to cover all the dementia in one lecture, therefore we should focus on the commonest kind of idiopathic dementia, that is Alzheimer's disease. In Alzheimer's disease, as you can see, the brain becomes atrophied. So the sulci and gyri become very prominent. And there is a progressive loss of neurons. In the MRI scan also you can see the ventricles are enlarged due to atrophy, and the sulci and gyri are so prominent because neurons have been lost. So what causes the Alzheimer's disease? A little bit has been known about the pathogenesis of Alzheimer's. There are certain genetic predispositions. For example, there may be mutations in presenilin 1 and 2 genes. There may be mutations in ApoE gene. Or there may be some unexplained or unknown causes. But whatever be the etiologies, what they ultimately cause is formation of an abnormal protein. So they lead to formation of an abnormal protein called beta 42. This beta 42 protein deposits in the brain. In extracellular space. And this deposit essentially forms an amyloid plaque, a plaque of extracellular proteins, also known as the neuritic plaques, which are huge at a microscopic level. These neuritic plaques cause a lot of different kinds of damage. On one hand, they stimulate free radical formation. On the other hand, they cause activation of protein kinase. The protein kinase, but the name tells you it causes phosphorylation of protein. It phosphorylates a protein known as tau into a phosphoprotein, tau phosphate. And these tau phosphates deposit inside the neuron. So remember, the amyloid plaques are extracellular, whereas tau phosphates are intracellular. And these deposits inside the neuron are known as neurofibrillary tangles. And these are relatively small in size because they are inside the cell. There is less space. The neurofibrillary tangles also lead to free radical formation, and probably they also lead to some mechanism that causes overactivity of the neurotransmitter glutamate. And overactivity of glutamate also causes free radical formation with the help of the NMDA receptor. So you see, ultimately we end up in the final common step, that is free radical formation. These free radicals damage different parts of the brain, but they particularly damage the cholinergic tract of Maynard. It's a large group of neurons in the brain that secrete acetylcholine, and that is responsible for memory. And this damage ultimately leads to loss of memory and dementia. So there are some genetic predispositions. There are some other etiologies. They cause formation of an abnormal protein called beta 42, and this beta 42 join to form an amyloid plaque, which are also known as neuritic plaque, which are very huge protein aggregates. These neuritic plaques cause protein kinase activation and phosphorylation of a protein known as tau. The tau proteins form neurofibrillary tangles inside the neuron, which are smaller in size. And ultimately, all these processes cause free radical formation. There is also some overactivity of the glutamate by some unknown mechanism, which accelerate the free radical formation. And they damage different neurons, including the cholinergic tract of Maynard, which causes dementia. Unfortunately, most of these causes cannot be reversed. So we have only two targets of treatment. We can supplement acetylcholine by inhibiting the metabolism of acetylcholine. So one target is to inhibit cholinesterase enzyme in brain so that acetylcholine remains in the brain, and this can be done by central cholinesterase inhibitors, and the drugs are donepezil and rivastigmine. The second aim is to reduce the excessive activity of the NMDA receptor, and this can be done with the help of a drug known as memantine. So currently we have two categories of drugs, the cholinergic drugs like donepezil and rivastigmine, and the anti-glutamatergic drug like memantine. And the treatment outcome is not very good. So the treatment is mostly rehabilitative. At the same time, these drugs are in trial. So this in a nutshell is the pathogenesis and management of Alzheimer's disease. Is it clear to everyone? So you see, this is a schematic diagram on the left hand side, which shows normal and Alzheimer's disease neurons. The large aggregates that are present outside are the amyloid plaques, also called neuritic plaques. And the small aggregates inside the neuron are the neurofibrillary tangles. Compare with this picture. Therefore, these large areas which are dark are the neuritic plaque or amyloid plaque. And the small dark areas within the cell bodies, these small dark areas, these are the neurofibrillary tangles. So the treatment includes central anticholinesterase inhibitors, anticholinesterase or acetylcholinesterase inhibitors like donepezil and rivastigmine, and NMDA receptor antagonists like memantine. So this is basically the end of the session. I kept it essentially very short because students from different levels are participating in this. Let us conclude by answering the questions. So this was one of the questions we started with. A patient with Alzheimer's will frequently complain that he is forgetting everything. Is it true or false? Very good. The answer is false. Next question. A potentially reversible cause of dementia is? Wonderful. The answer is AIDS dementia. Example of multifocal dementia is? Very good. The answer is prion diseases. So what is this labeled structure? So the label structure is large and the small black areas are actually the cell bodies. So this huge area must be present outside the cell and therefore that should be an amyloid plaque. Therefore it is a neuritic plaque. So the answer is B, neuritic plaque. Finally, the treatment of Alzheimer's disease includes? Yes, the answer is all of the above.